our speaker is an emeritus professor in clinical medicine and an honorary consultant physician to the National Hospital of Sri Lanka. He is an alumnus of the Faculty of Medicine, University of Colombo, and joined the staff as a lecturer in 1982. He got his MD from University of Colombo and University of Bristol, and also he is a fellow of the College of Chest Physicians and a fellow of the Royal College of Physicians, UK. He was also the founder director of the Medical Education Development and Research Center. He was the former chair professor of medicine in Faculty of Medicine, University of Colombo, and co-organizers an interdisciplinary postgraduate diploma in health development in the university. His research interests include innovations in clinical teaching, health equity, social determinants of health, and application of complexity science to clinical medicine and population health. So please join me in welcoming Emeritus Professor of Medicine, Professor Saroj Jayasinghe. Over to you, Professor Jayasinghe.
Thank you very much. Once again, thank you for inviting me. Can you hear me? I think the slides are coming up now. Yes. Okay, I think my image is not coming up well, but you've seen me, so I'll take my image off the screen. Can you all hear me now? Is everything clear? Gayani?
Yes, Professor, yes.
All right, okay, right. So, yeah. So my topic is on medical devices, doing the science right. And I have looked at this. I begin with a small story on endothelial dysfunction and then get on to apply what we've learned from this to a bit of theory in relation to science of device development. So this whole episode began somewhere, I think it was about 2015, when a young senior lecturer from Moratua, Anjula De Silva, who walked into my office, and with him there was Mr. Kirti Kodituaku, who was a student at that time, and he wanted to do some studies in relation to biomedical devices. And we got chatting, and somewhere down the line, we came up with this idea of trying to look at the endothelial function. Now, endothelium is, in fact, the cellular layer inside all blood vessels. And though it's just one single layer, there are a lot of functions related to it. And there was data that it acts as a precursor, or it predicts future atherosclerosis. So future deposition of cholesterol in the lining of the blood vessel seems to be related to the function of this inner layer of cells. And with more and more research, they found that previously we thought this was just a cell layer, but this cell layer produces a lot of compounds, including nitric oxide and various other compounds, which have an effect on the health of the blood vessel, and in fact releases certain compounds which have an impact on the functioning of tissues also. So it looks as if this endothelium, this single cell layer inside blood vessels, is crucial for us. When you look at diseases such as diabetes, we found that even there, the changes in the metabolism of blood glucose has an effect on endothelial dysfunction. So once the idea came about, the science of it was something which was already published, and that's probably one of the most important steps, because you have to look around and see whether your idea can be validated, whether it's reasonable based on the what is known about the human body, and whether other studies have been done. And with that came once this idea was shared, we found one or two other publications, one from Israel and one from Russia, which where they had used an instrument. And of course, Anjula and Kirti, being brilliant in the areas, they developed this device, which basically uses the pulse oximeter, which we use commonly in the wards, and through a set of web-based software and machine learning, etc., which is able to look at the blood flow or give an index of the blood flow in the vessels of the fingertips. Now, the science of it is based on this change in the pulse wave we see with each heartbeat. So the sensors here are in fact detecting this pulsatile arterial blood flow at the fingertips. Then the other area which was looked at was the effect of having a cuff. When you have a cuff here above the elbow and you check the blood flow, here there is a probe here, but you have the probes here also. You keep it closed for some, you inflate the cuff so that there is no blood flowing to the arm. And you can do this for a bit of time, nothing happens to your arm. And then when you observe the blood flow here, you find that the blood flow will fall. And how do you know that the blood flow falls? Because you find that the temperature falls. And you release the cuff, and then the blood flow starts to come back. And in fact, it overshoots. So it dilates. Now, this dilatation, this opening up of the blood flow is related to the things which are produced in the endothelium. So if we know that when you occlude, there is what we call a hyperemia, that's an excess of blood, you can look at these waveforms and see whether the endothelium here and the nitric oxide, etc., which is produced here, whether the endothelium is reacting properly. Okay, so that's again the science of it. So that's where you use the digital temperature and you use the digital thermal monitoring, and you are trying to see the rebound which happens as an index of the blood vessel opening up. So if the blood vessel is diseased, it won't open up the way it should. If it is functioning normal, it should open up. So that's part of the scientific approach, is to read what is available, know what's happening, and learn the mechanism. And you'll need expertise from the medical field, if it's in the medical field, to try and explain this phenomenon. The next stage was, of course, to see whether, once this was produced, whether this would have some effect on whether we can measure it the way other machines are measured. And all medical studies require ethics approval from ethics committees. There are recognized ethics committees. Now, University of Moratuwa also has, but there are certain ethics committees which are well recognized internationally, like the University of Colombo Faculty of Medicine. And we submitted this to the Faculty of Medicine, Colombo, to see whether we could look at some preliminary data. And we did that in the National Hospital. So we looked at people who are normal and compared it with people who had obvious vascular disease. So people who had advanced vascular disease, we knew from their tests, their ECGs, their past history of what they've said, and investigations, for example, coronary angiograms and diabetes and so on. So we wanted to see if there was a difference between normal and those who were at that stage. It was done using a private sector investor, and it was the Asari Group of Hospital, its own ethics committee. And again, here we looked at people who were normal, and you compare with people who had obvious vascular disease. Their blood vessels had atherosclerosis. We had three bright medical students who did the electives with Kirti's team. Priyankara is Kirti. And they they wrote this paper on whether this could be used of severe COVID, because we know COVID, when it is severe, has problems with the vessel wall. And similarly, we are thinking of dengue also, because dengue also we know that the vessel walls are affected. That's why the blood pressure drops and so on. So this study is to be done. The concept paper has been written. We've started to plan for this study. And this shows the progress of the science of it, because once you do the pivotal study, and if it is accepted by the regulatory authority in Sri Lanka, it's NMRA. And if you Google and go to NMRA, you will see a separate section for devices. And they have a special committee which looks at medical devices. And that special medical committee will want studies, for example, the pivotal study, which I was mentioning. We have to do more. It's only a small number in that research paper, but we have to do 300, probably 300 or 400. And then we have to get approval and then start using the device in different areas. And one area is, of course, try and see whether this can be used in the COVID epidemic. But long-term studies have to be done. So that's the story. Now, why I told you a story, a real life experience, is that you remember the steps. But now let me dissect these areas which we covered. So the science of device development. So what can we learn from this experience, and also what can we learn from the literature? So firstly, I gave you an example of a vascular device, device used for some vascular, to look at vascular health. But there are lots of devices which we see. What are the scientific mechanisms of the devices, or what the device you are trying to propose, and what are the scientific studies you have to do? So that's all. This forms part of the science behind a medical device. Now, when you look at devices, we used a fairly easy device because there is no invasiveness. It's just a probe, very similar to the pulse oximeter, which is used very, very commonly even in the ICU. The difference was that here we had to apply the cuff and release. So when we submitted to the ethics committee, they were very concerned about that, because you're having a cuff in an ill person, you're going to have a cuff. So that's relatively simple. device because it's outside the body. But there are more important and interventions through devices. Right? So, for example, you can have devices onto the brain, stents, then joint devices. Uh, Someone has got the mic on and there's a noise coming up. Right, okay. So we have these devices which are invasive. And that's tricky, isn't it? Because it's obvious. If you are going to insert foreign bodies into into a human, you will have a lot of more research required, much more science than just a device like a pulse oximeter. Now, there are certain devices where you don't have to actually do any such things because they're external. For example, a wheelchair. Again, a device. You can have an electrical wheelchair and so on. You might have devised a ECG machine. So you don't expect people to have any sort of side effects due to the the ECG. So the important thing is that there are several definitions of devices, and we have classes of devices also. So if you try to really define what a device is, it's an instrument, apparatus, appliance, and so on. So many things. We will describe it further. And it's used to diagnose, prevent diseases, monitor, treat sometimes. For example, pacemakers, you treat when there's a heart rhythm abnormality. You can have devices to investigate, like the device which we spoke about. You can have devices to replace function, like a hip replacement, right? And it affects the structure of function. It's not just a device which you keep, but there is some structure or function. Even there are silicon implants for breasts. That's also a structural change, and that's considered a device. But the crucial difference between this and most other treatment modalities is that it's not through chemical action. But now there is an overlap because we have stents. You put a cardiac stent, and the stent is releasing certain compounds to prevent the clot forming, right? So there is some action, but the primary thing in a device is that there is no chemical action of a device. Okay, it's the same what I've explained. Okay, and then you get these. I briefly mentioned about the classes of devices, things which are external, like gloves. Also, you can think of them as a device, but they have general controls of quality and so on. And they are most exempt from pre-market submission. But for example, the masks which have been produced now, different types of masks, we have seen certain universities, I think Peradeniya, coming up with a new mask and so on. Then Slintech came up with so many things with the COVID epidemic. Then you have these class 2 medical devices where some special controls, like the instrument which we developed, which Kirti and Anjula and them developed. There are certain special controls of the pulse oximeter you have to see. You have to make sure that it works properly. You have to make sure that it is valid and so on. So that requires certain studies. Before we market it, you have to do your pivotal study, and NMRA has to okay the studies. Studies as such, like this. But when it comes to class 3, like a stent here, right, you will need approvals, stronger approvals, and they have to be done in a very scientific way. So if you look at the science behind it. So I described the types of devices, then the science behind it. Firstly, you must know the scientific mechanisms. So if you are developing a device, you will have to start reading up or meet the scientists, meet the medical specialists and discuss, is this a good idea? How do I take it to the next level? And then you have to think of the studies which I described with the with the endothelial dysfunction uh, device. And these are the types of studies. Sometimes you do certain studies to see whether your idea is working. It might be a device, that's the sort of device which is produced in the final year projects by the students, where you proof of principle and first in human studies. Okay, but those are not so important uh, for registration purposes, because you have to go beyond that. You have to go beyond that when you want to get approvals to use it. And here you require pilot study. Right? Now, even with the study which involved the device for endothelial dysfunction, there are safety issues. Could there be a electrical short circuit and so on? Then we have to get on to pivotal studies. That's the crucial one, which is required by regulators. So there are large number of people have to undergo the studies. And like in drug trials, after that you have to look for post approval. That is, once it is in the market, will it really do what it's supposed to do? So will the machine really help to detect people who are at high risk of cardiovascular disease? And these long-term effects you can further classify on the performance. That is, does it really achieve the intended purpose? And if it does indeed assesses the vascular endothelial dysfunction, does it really help to or is it sensitive enough and specific enough to predict cardiovascular disorders? And then, if by detecting cardiovascular disorders early, does it really cause a benefit? I mean, if you're going to predict the cardiovascular benefit by doing just the serum cholesterol, why bother with this device? You might as well just do the serum cholesterol and act accordingly, right? So you can look at the long-term effectiveness also from different dimensions. And when it comes to things which you insert into the body, it becomes still more complex, because these preclinical studies become very important. Okay, so this is almost the same what I described in relation to the endothelial dysfunction studies. But if, for example, you have developed a stent, or you have developed some implant, then you will see you will require more studies. Sometimes studies in relation to dead bodies. Can you insert it? How can you insert it to a human body? Can you do it in animals and try? I mean, a lot of animals around the world are killed because of devices, and they're tortured, I think. But anyway, that happens. And some of these devices are inserted into animals. And of course, what are their safety in relation to inserting them? And now this device development has gone to a different field. Some of these can be studied by computer simulation. And once you get the device, you can 3D print it. So things have really changed now. And it's becoming an accelerated process. Now, just a brief description on device studies compared to drug studies. You see, the medical profession is quite used to this. When you find a new drug, how you test it out, right? So phase 1, phase 2, phase 3, phase 4, there are very strict criteria on how we do that. But devices, it's slightly different the way we do studies. Because, for example, in drugs, you have this double-blind control study, randomized double-blind control study, where the patient doesn't know whether it's a drug A or drug B the person is taking. The person who prescribes and gives the drug also doesn't know because it's in a code. So he or she also doesn't know whether it's A or B. When it goes to the statistician, the results go to the statistician. Statistician also doesn't know whether it's A or B. They just group it into group A, group B, and do the statistics. Once everything is done, then they break the code and say, right, A is the new drug, B is the old drug or the placebo. So that's a double-blind trial. Now you can't do that with a device because the device is something the person sees. Very, very rarely there are sham devices which we put in onto people, but that's very rare. So devices, the studies are different. The types of studies we do are different. The sort of observations we do are different. The side effects, the complications, and so on. And the process used is therefore different. So in summary, doing the science right, I told you a Sri Lankan story on endothelial dysfunction. I hope you remember the steps because how the idea came about, how we explored the idea with literature and looked at the scientific mechanisms, and how we did the initial study was done in Morocco itself. Then the pilot studies, how we did it at the National Hospital, and then how we got on to, and we are now doing the pivotal studies with a private sector investment on looking at a large group of patients. And finally, we'll get on to the post-approval efficacy of performance studies. So once again, I must thank the organizers for inviting me. And also, certainly, I must acknowledge Angelo De Silva and Kirti Kodituako and his team at Gendo and everybody else who has supported me and been part of this very, very interesting field of study. Thank you.